Hi, my name is Carole Lunny, and I'm a postdoctoral researcher at St. Michael's Hospital in Toronto and the Cochrane Hypertension Group at the University of British Columbia. I'll be talking today on behalf of our co-authors about our methodological review of items, which will provide the groundwork for the development of a new risk of bias tool for network meta-analysis. Please tweet this presentation at Carole_Lunny and use the hashtag NMA. So we have received funding for this project through a 2021 CIHR project grant. We have no conflicts of interest to declare, and we wish to acknowledge the traditional ancestral and unceded territory of the Musqueam, Squamish, Dolo, and Tsleil-Waututh nations on which the research team operates in Vancouver, BC. Our project team consists of the steering committee, our stakeholders, and the working group. The steering committee consists of myself, Julian Higgins, Ian Wright, Sofia Diaz, Andrea Tricco, Brian Hutton, Argie Veroniki, Jim Wright, and Penny Whiting. We're also working with students and an international group of stakeholders who will help us in our dissemination efforts. As a background, so NMAs can compare all treatments for a condition, whereas traditional meta-analyses are limited to comparing only 2 treatments or interventions. Network meta-analyses provide evidence to clinicians faster than traditional meta-analyses due to the lack of evidence from head-to-head comparative trials. For example, researchers recently showed that prostaglandins, seen here on the left, would have been identified 7 years earlier as the most effective drug class in lowering intraocular pressure for open-angle glaucoma if an NMA had been done at that time. NMAs help patients and their care providers choose the treatment that is most important to them based on efficacy and side effects of all treatments. So why is it important to assess NMAs for biases? Without a risk of bias tool for network meta-analysis, knowledge users will not be able to tell which NMA is at low risk of bias. Many NMAs are published on the same topic, and choosing from conflicting NMAs is impossible without a risk of bias assessment tool. In the study on the right, for example, the authors found 28 network meta-analyses on the treatments for rheumatoid arthritis, all with differing and conflicting results. So if an NMA is at risk of bias and inappropriate methods are used, the validity of the findings can be compromised, which can impact on treatment and patient treatment and care. So what do authors mean when they say they're assessing the quality of a study? So when you read in a study that the authors assess the quality of the included studies, do they mean that they assess the quality of reporting? Do they mean how well the study was conducted, or whether the study was at risk of bias? Sometimes we just can't tell. So reporting quality is about how well the methods and results in a published study are described. Methodological quality is about how well the research is conducted. And risk of bias is about whether the limitations in a study can potentially bias the methods, results, and conclusions. So bias refers to systemic error, meaning that multiple replications of the same study would reach the wrong conclusion on average. So bias should not be confused with imprecision. Imprecision refers to random error, meaning that multiple replications of the same study will produce different effect estimates because of sampling variation. For example, the results of smaller studies with few events are less precise and produce wide confidence intervals. Bias is also not the same as quality of conduct in a study. Even a well-conducted study can be at risk of bias, for example, in studies where blinding is impossible, for example, in surgical trials where the surgeon can't be blinded. Similarly, not all methodological flaws are relevant to bias. For example, failure to perform a sample size calculation or to obtain ethical approval are all important markers of study quality of conduct, but they're unlikely to bias study results or conclusions. And finally, bias is not the same as the quality of reporting. So studies rarely report the methods that they used in great detail. And studies may have been rigorously developed methods, but they might not have described those methods in the published paper. So this table shows that several tools are designed with different purposes, some for assessing reporting quality, some for assessing quality of conduct, but currently none are designed to assess risk of bias in network meta-analyses. For critically appraising the quality of reviews, the AmStar2 checklist is the most commonly used tool and aims to evaluate the quality of conduct of reviews. Robust is a domain-based tool used to assess the risk of bias in systematic review conclusions. And to assess reporting quality of network meta-analyses, users could use the Prisma NMA statement or the NICE DSU checklist. To assess quality of conduct in NMAs, users can use the 2014 iSport checklist. However, due to important methodological advances in the field of NMA, the iSport checklist, which was published in 2014, is currently out of date. As I want to emphasize, there's still no risk of bias tool for network meta-analyses. So our objective and methods. Our objectives are, first of all, to conduct a methodological review to identify items related to biases in NMAs for potential inclusion in the tool, which is the focus of this presentation. Our further objectives are to conduct a multi-round Delphi survey with experts who will make their selection of items on which to include in the tool. We also want to conduct a survey to ask stakeholders about how they'll use our tool. We want to pilot test our tool for usability, efficiency, comprehension with different user groups. And finally, we want to develop and evaluate a knowledge translation and training plan for tool dissemination. So my co-authors and I developed a research protocol, which is found as a preprint on Research Square and is currently under consideration with BMJ Open. For our review, we included journal articles, guidance documents, and methods papers that describe items used to assess bias in NMAs. We searched Medline, the Cochrane database, methods collections, and the Gray literature. We extracted data on items that are potentially relevant to bias. And items were extracted and analyzed by 2 authors independently and discussed until consensus was reached. We grouped similar items. We split items so that each item covers a single concept during data analysis. And finally, items were reworded as signaling questions so that each item was phrased so that yes is considered good. So what did we get as our results? So after screening and data extraction, 63 studies were included, which yielded 99 items. After ordering and refining, a final list of 22 items were retained by the steering committee, and these items will be entered into a Delphi survey for experts to get their feedback on whether items should be included or excluded. So in conclusion, our methodological review of items provides the groundwork for the development of a new risk of bias tool for network meta-analysis. We will conduct our Delphi and stakeholder surveys this summer to ask for expert feedback and knowledge user feedback to further refine the items in the tool. And finally, knowledge users will be able to evaluate network meta-analyses for important biases in their evidence-based decision-making, thereby increasing the uptake of relevant evidence and ultimately improving patient outcomes. I thank you, and I welcome any questions.